In today's video, we're going to be taking a look at the sliding filament model for A-level biology. Now, in order to understand how we get calcium ions present in the sarcoplasm, I'd recommend taking a look at my video on the neuromuscular junction. Otherwise, let's go ahead and get started. So here we have our myosin filament, right, with the bulbous head that protrudes, and then we have the actin filament. Now, the actin filament is associated with two proteins. Number one is tropomyosin, which is composed of two long polypeptide chains that wrap around one another, right? We can see that wrapping around. That's the tropomyosin. And then we also have the troponin molecule, the troponin protein, which is a globular protein that attaches to tropomyosin, forming a troponin-tropomyosin complex. Now, the tropomyosin blocks the binding sites on the actin molecule, so the bulbous head cannot attach to form a actomyosin link, a cross bridge. So what then happens is those calcium ions attach to the troponin, which will then cause the troponin-tropomyosin complex to change shape, opening, right, making available the binding sites of the actin molecule. What will then happen is the bulbous heads will attach to the actin binding site, therefore forming an actomyosin cross bridge, as we see here. What will then happen is the bulbous heads will do a power stroke. So here we can see that the bulbous head had this shape, it attached. Then after the power stroke, it has now moved, and in doing so, it has slid the actin filament along the myosin filament. Now, it's very important to understand here that we're just taking a look at the action of one bulbous head from a myosin filament, right? In reality, there are going to be numerous. There are going to be so many myosin heads doing this action, but right now, for explanation purposes, it's just with one myosin head. So that myosin head will do the power stroke, and then ATP will attach to the bulbous head, releasing, right, detaching the bulbous head from the actin binding site, and then the ATP aces within the bulbous head will break down, right? They'll hydrolyze the ATP into ADP, an inorganic phosphate, which will release energy, and that energy will then be used to recock the bulbous head back into its initial position, where it can from then attach to another actin binding site. Okay, and we will have that ADP that is attached to the bulbous head until it is attached, done the power stroke, and then ATP again binds to it in order to release it from the actin binding site in order for it to return to its normal position. Now, this is all muscle contraction, and how this will actually result in muscle contraction will go into more depth in the video on striated muscle. But what happens in muscle relaxation is that if there is no nerve impulse being transmitted, right, which we saw in the neuromuscular junction video, then the sarcoplasmic reticulum will actively take up calcium ions, and so the tropomyosin will block the binding sites on the actin molecule, on the actin filament, and so this power stroke will not be able to take place, so muscle contraction won't be able to take place. So that's it for this video. If you found it helpful, I'm very happy. Take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thanks very much for watching, and I hope you have a great day. Bye-bye.